As lymphocytes differentiate from primitive stem cells, they proceed through stages that are marked by the sequential rearrangement of the antigen receptor gene segments at the different genetic loci. As each complete receptor chain gene is generated, the protein it encodes is expressed as part of a receptor, and this signals the developing cell to progress to the next developmental step. For B cells, the heavy chain locus is rearranged first. If rearrangement is successful and pre-B cell receptor is made, heavy chain gene rearrangement ceases and the resulting pre-B cells proliferate, followed by the start of rearrangement at a light chain locus. If the initial light chain gene rearrangement is productive, a complete immunoglobulin B cell receptor is formed. Gene rearrangement ceases and B cell development proceeds. If it is not, light chain gene rearrangement continues until either a new productive rearrangement is made or all available J regions have been used up. If a productive rearrangement is not made, the developing B cell dies. In developing T cells, receptor loci rearrange according to a defined program similar to that in B cells. There is, however, the added complication that individual precursor T cells can follow one of two distinct lines of development. There are 4 T cell receptor gene loci: gamma, delta, alpha, and beta. Rearrangement at these loci leads to cells bearing either gamma-delta T cell receptors or alpha-beta T cell receptors. Early in ontogeny, gamma-delta T cells predominate, but from birth onward, more than 90% of thymocytes express T cell receptors encoded by rearranged alpha and beta genes. In developing thymocytes, the gamma, delta, and beta loci rearrange first and start rearranging virtually simultaneously. Productive rearrangements of both gamma and delta gene may lead to the production of a functional gamma-delta T cell receptor and the development of a gamma-delta T cell. Most cells, however, enter the alpha-beta lineage. In these cells, the generation of a beta chain gene before both gamma and delta have rearranged leads to the expression of a functional beta chain and a pre-T cell receptor. This receptor, like the pre-B cell receptor, signals the developing cell to proliferate and to arrest beta chain gene rearrangement, to express CD4 and CD8, and eventually to rearrange the alpha chain gene. Rearrangement of the alpha chain genes continues in these CD4+, CD8+ double positive thymocytes until positive selection allows the maturation of a single positive alpha-beta cell. Almost all cells will make productive alpha gene rearrangements, but the vast majority of thymocytes die at the CD4, CD8 double positive stage because they are not positively selected. Thus, gene rearrangement follows an ordered sequence in both B and T lineages to produce immature lymphocytes that each bear antigen receptors of a single specificity on their surface. These antigen receptors can now be tested for their antigen recognition properties and the cells selected accordingly. Have a great day.